Hi, my name is Prabjot,
And my name's Ravni.
and welcome to today's episode of BioBlast. Today we'll be talking about the world-famous Human Genome Project.
We will begin with the basics and move on to the history of the Human Genome Project.
We'll be discussing the political, economical, and sociological effects of the Human Genome Project in this episode.
So let's begin. So what is a genome? A genome is an organism's complete set of DNA, including all of its genes. Each genome contains all of the information needed to build and maintain the organism. In humans, a copy of the entire genome, more than 3 DNA base pairs, is contained in all cells that have a nucleus.
Gregor Mendel was a key individual in the history that led to the famous Human Genome Project. He was the first to undertake a series of brilliant hybridization experiments with garden peas, which led him to create the laws of heredity and genetics. His experiments included the crossing of pea plants to see how traits were passed down. He observed specifically different heights, colors, pod shapes, and seed shapes. He experimented with the peas from 1856 to 1863, and in 1865 he published his work revealing that genes determine traits.
Friedrich Miescher collected bandages from nearby clinics and washed off the pus on them. By doing so, he discovered that the nuclei, made of hydrogen, oxygen, and nitrogen and phosphorus, is within a cell's nucleus. He believed that proteins were the hereditary molecule within humans.
Rosalind Franklin. Now, who is she? Well, she played a major role in discovering the shape of DNA, a double helix, with the 2 helixes running anti-parallel. She used the technique of X-ray crystallography.
So Watson and Crick were the first to visually compose a structure of DNA with the aid of Franklin's research. Their model represented DNA as a double helix, with sugars and phosphates forming the outer strands of the helix and the bases pointing into the center. Hydrogen bonds connected the bases, pairing A and T, C and G, and the 2 strands of the helix were parallel but oriented into opposite directions. This unveiling was one of the biggest contributions to the scientific world, as it is the base component of understanding DNA.
Marshall Nernberg. Marshall and his postdoctoral fellow, J. Henrique Mathey, conducted an experiment which helped them crack the genetic code for protein synthesis. They used a sample of bacterial cells which could make protein without any living cells. To this sample, an artificial form of RNA was added, poly-U. This helped discover the first codon of the genetic code and concluded that RNA played a major role in controlling the production of which types of protein were created. After a couple of years, they discovered the entire genetic code by matching amino acids to specific triplet nucleotides. A redundancy in the codons and stop codons were also discovered.
So, who was Frederick Sanger? Frederick Sanger is the one who developed the DNA sequencing technique in 1977. It is a process of determining the order of bases in a strand of DNA.
In 1990, the Human Genome Project began with the NIH and DOE developing the plan for the first 5 years of a 15-year plan. The plan discussed to develop a technology for analyzing DNA, mapping and sequencing human and other genomes, and studying ethical, legal, and social issues. The private sectors also played a significant role in the project. So specifically, the development by the Applied Biosystems Inc., who are responsible for automated capillary sequencing that generates data for the first human genome sequence. However, the project became international as more than 18 countries also took part in it.
There were 4 major goals established for the project. First, to identify all the approximately 50,000 genes in the human DNA. Second, to determine the sequence of 3.2 billion base pairs that make up the human DNA. Third, to store this information in databases. And lastly, to create tools for data analysis and discover any ethical, legal, or social issues that may arise.
When the project was first proposed in the mid-1980s, one prominent skeptic was Nobel laureate David Baltimore, who estimated that it would take 100 years to sequence the human genome, and many agreed. The 100-year estimate would in fact have been about right if the technology had remained frozen at the level of 1985. But the technologies used in sequencing the genome improved rapidly, and the century of work was compressed into 15 years.
Another eminent geneticist, David Bornstein, warned less seriously at a national meeting in 1986 against becoming involved in the mindless big science of sequencing genomes.
Baltimore and Bornstein both quickly recognized the value of the project and were active in gathering support. They are examples of how good interactions and scientific debate in the community informed and built the case for the Human Genome Project even in the face of broad opposition.
One of the first goals accomplished of the Human Genome Project was the sequencing of the genome bacterium Haemophilia influenza. It was sequenced with shotgun sequencing, which is the use of computable softwares to assemble short overlapping DNA fragments without any preliminary physical map of the genome. This method was then later used for the entire sequencing of the other genomes within different organisms.
Perhaps the most important industry role in the Human Genome Project was that of Celera, a private company led by J. Craig Venter, which was a key part of the endgame of the project. Although there were ultimately 2 genome projects, one public, including international efforts, and one private, there was substantial mutual benefit. The Celera effort was initially seeded by Doe's Office of Health and Environmental Research, but the effort was organized, funded, and led independently of the public project through many scientists with long involvement in the project contributed by joining the Celera effort or becoming advisors. In retrospect, the Human Genome Project is not likely to have been completed as early as it was without the massive effort by Celera using some newly devised methods.
The public and private projects use similar automations and sequencing technology, but different approaches to sequencing the human genome. The public project uses a hierarchical shotgun approach in which individual DNA fragments of known positions are subjected to shotgun sequencing, i.e., shredded into small fragments that are sequenced and then reassembled on the basis of sequence overlaps. The hierarchical shotgun method has the advantage that each individual sequence's global location is known with certainty. However, it requires constructing a map of a large fragments covering the genome. The whole shotgun method does not require this step, but possesses other lacking qualities. On the other hand, the Celera project uses a whole genome shotgun approach in which the entire genome is shredded into small fragments that are sequenced and put back together on the basis of sequence overlaps. Thanks to the Human Genome Project, we now know that our genome contains about 3.2 billion A, C, G, and T bases, the molecules that help us make up the DNA code. An average-sized gene has about 3,000 of these bases, though some genes are smaller and some are much bigger. The biggest known human gene, dystrophin, is made up of about 2.4 million bases. The Human Genome Project also gave us more detailed information about chromosomes. It turns out that chromosome 1 contains the most genes, 3,168, while the Y chromosome has the fewest, of about 344. In February 2001, scientists published their first interpretation of the human genome sequence. They discovered that humans possess only 30,000 genes. This discovery led to scientists to realize that human complexity does not come from the quantity of genes, but from the network of different genes, proteins, and groups of them.
The first technique used was DNA sequencing. This was important because it was used to find the functions of the genes as it determines the exact sequence of bases in a DNA molecule. The second technique used was the polymerase chain reaction. This was used to amplify DNA sequences. The third technique used was electrophoresis. This was used to separate DNA, RNA, and proteins based on different sizes. The fourth technique used was the employment of restriction fragment length polymorphisms. The fifth technique used was yeast artificial chromosomes. This is an artificially constructed system which allows for replication of large segments of genetic material, known as cloning. And the last technique used was bacterial artificial chromosomes, BAC. The first benefit is molecular medicine. Now, because of the Human Genome Project, doctors can worry less about symptoms of new medicines because they'll be able to know what genes are related to what diseases. Also, doctors will be able to prevent diseases prior to possessing them due to the knowledge of what genes are linked to what diseases and if the disease is hereditary. Comparative genomics. Due to the Human Genome Project, able to compare human genomes with other forms of life and properly understand human evolution and how humans are connected to other forms of life. Genetic archaeology is also being developed and becoming more common in today's society. The Human Genome Project is also beneficial to forensic science. This is because due to the Human Genome Project, you can create a DNA profile of a person. This allows police to identify suspects whose DNA may match evidence left at a crime scene, and can also be used to prove others innocent who are wrongly accused or convicted. Due to the Human Genome Project, farmers are able to have better crops and animals. Having a stronger genetic understanding of plants and animals allows farmers to grow crops which will be able to resist diseases, droughts, insects, and so many other factors.
By this, it will help farmers produce healthier and stronger livestock, fruits, and vegetables.
So that concludes today's episode of BioBlast. Hope you understand the significance of the Human Genome Project and the benefits that resulted thereof.
See you next week for another episode on the telomeres.
Bye!